Could memory loss actually be reversed? Scientists at the University of California, San Francisco, have made a groundbreaking discovery that could change how we think about age-related cognitive decline. Their research, conducted on mice, reveals a key protein in the brain that drives memory deterioration with aging, and even more exciting, they found a way to dial down this protein to restore memory function in older animals. But here's where it gets controversial. Does this mean we are on the brink of a true cure for dementia, or are we merely scratching the surface of a much more complex puzzle? Worldwide, the incidence of dementia is expected to triple by the year 2050, creating a looming public health crisis. Unlike Alzheimer's disease, normal aging doesn't typically involve massive brain cell death, but it still results in frustrating memory lapses, like forgetting where keys are placed or struggling to remember names. Understanding the underlying causes of these changes has remained elusive until now. The research team focused on a protein called ferritin light chain 1, FTL1, which accumulates in the brains of aging mice. This protein is involved in iron storage within brain cells, and as mice grow older, FTL1 levels spike, correlating with worse performance on memory tasks. Intriguingly, when FTL1 was artificially increased in young mice, their memory quickly declined, mimicking the cognitive impairments seen in older animals. Conversely, reducing FTL1 in elderly mice led to remarkable improvements in their ability to learn and remember, effectively turning back the clock on their cognitive abilities. What's happening at the cellular level? FTL1 appears to disrupt how brain cells produce energy. Neurons require a robust energy supply to maintain connections critical for memory formation and recall. When FTL1 accumulates, it hampers mitochondrial function, the cell's powerhouse, resulting in reduced energy and weakened synaptic connections. By supplementing the mice with NADH, a molecule that boosts cellular energy production, researchers were able to offset FTL1's damaging effects and improve cognitive performance. This discovery is particularly provocative because it links iron metabolism in the brain directly to memory loss, an area that has been underexplored. Although elevated ferritin levels in human spinal fluid have been linked to faster cognitive decline and a higher risk of Alzheimer's, FTL1 itself hasn't been extensively studied in people. Given that FTL1 is present in human brains, these findings open the door to potential new therapies aimed at preserving cognitive function as we age. However, there are important caveats. The study included hundreds of mice, but exclusively male specimens, raising questions about whether these effects hold true for females. Moreover, while the hippocampus, the brain's memory center, was the focus, it's unclear how FTL1 behaves in other regions of the brain. Long-term safety and the implications of manipulating FTL1 remain unknown, and direct application to human dementia treatment is still far off. The researchers conducted their experiments by comparing young adult mice, equivalent to roughly 20-year-old humans, with older ones, similar to people in their 60s. Using sophisticated genetic tools, they precisely increased or decreased FTL1 levels in hippocampal neurons and then tested memory using tasks like the Y maze and novel object recognition. Consistently, reducing FTL1 in aged mice restored not only memory, but also synaptic connectivity, offering hope for genuine cognitive rejuvenation rather than merely delaying symptoms. This bold discovery forces us to rethink, could targeting iron metabolism become a key strategy in combating age-associated memory deficits? Or might interfering with these delicate processes carry unforeseen risks? What's your take? Do you see this as a breakthrough in reversing dementia, or just an early step on a challenging road? Share your thoughts and let's discuss. Summary. FTL1, a protein regulating brain iron, builds up with aging and impairs memory in mice. Increasing FTL1 in young mice causes memory problems. Reducing it in aged mice improves cognition. Excess FTL1 disrupts neuronal energy production, weakening memory-related synaptic connections. Supplementation with NADH, which supports energy production, reverses these cognitive deficits. The study was done exclusively in male mice, so further research is needed to confirm effects in females and humans. These findings highlight FTL1 as a promising target for future therapies addressing cognitive decline. This exciting research, funded by multiple esteemed foundations and institutions, was published as a letter in the journal Nature Aging in August 2025. The team is led by Laura Remisal and Saul A. Valletta of UCSF, with collaborators from other major universities across the U.S. Their cutting-edge work may one day pave the way for new interventions to maintain our memories well into old age.